All right, in this video, I am going to go over a quick crash course in genetics, mostly because you need to have a good understanding of genetics to know what's going on with evolution. Because really evolution, you're looking at genetics at a population scale. So if you don't understand what's happening at the individual level, it's going to be really tough to understand what's going on at the population level. So again, this is just really fast to give you a quick background so you can move on to the evolution part. All right, with this, since I'm going to keep it simple, I am just going to talk about eukaryotes specifically. So if you don't remember what a eukaryote is, you might want to go back and just refresh your memory on the difference between eukaryotes and prokaryotes. With eukaryotes, we're also going to talk about ploidy. And then after we're done talking about ploidy, I'll go in a little bit about the difference between genes and alleles. And then after I talk about that, then we'll talk about genotypes and phenotypes. All right, but just to get started, let's do a quick refresher on what genes are and how they apply to the traits that you express. All right, so with genes, when we're talking about a gene, what we're really talking about is a sequence of nucleotides in your DNA that are going to code for a protein. All right, so you have genes which are made up or found in your DNA, and all DNA is long strands of nucleotides. Now you have four major nucleotides in your DNA. You have adenine, thymine, guanine, and cytosine. All right, so these are the four molecules that will make up your DNA. All right, and the genes that you have, the traits that you have that you express are all based on the sequence that these four nucleotides come in your genes. So you might find a gene somewhere that has this certain sequence of nucleotides, and it will code for a specific protein, which will then give you that trait. And this will lead us to what is known as the dogma of genetics. And by the dogma, we mean how we get from a DNA coding to whatever trait you are expressing. And this is done in 3 steps. So you have your DNA to begin with. So whatever strand of DNA you have in your gene, or whatever strand of nucleotides you have in your gene, those are going to be transcribed, meaning copied, into RNA. RNA is what's going to leave the nucleus, and then an RNA is another sequence of nucleotides that is going to be translated into proteins. And the proteins are really what do the work. So this is what's going to give you your trait. So all the traits in your body are either made from proteins or made up of proteins. All right, so this again was called the dogma of genetics. Okay, so now we're going to talk about ploidy, genes, alleles, genotypes, phenotypes, stuff like that. And to do that, we're going to focus on the human genome as an example, mostly because I'm biased, because I am a human. So the first thing we're talking about is chromosomes, which is how DNA is organized in your cells. All right, with human genome, what I mean by genome is all the genes found inside the nucleus of your cell. All right, and every cell in your body has the same genome. You have 37 trillion cells in your body, and they all have the same genome, meaning they have the same genes and the same sequences to those genes. But with the DNA inside your genome, it's not just one long strand. So your DNA is not going to look like something like this in every single cell. Instead, it's broken up into smaller strands called chromosomes. So this is more how DNA is going to be found in your nucleus, in your cells. All right, so they're broken up into chromosomes, which are strands of DNA. Now, depending on the organism we're talking about, you could have different numbers of chromosomes. And with humans specifically, we have 46 chromosomes total. All right. Now your chromosomes come in pairs, and I'll talk a little bit about what we mean by pairs of chromosomes here in a little bit. But since they come in pairs, and you have 46 total, that means every single nucleus is going to have 23 pairs of chromosomes. Now, whenever a cell has pairs of chromosomes, we have a word for that. All right, we call it diploid. So now we're talking about ploidy specifically. By ploidy, we mean, do you have pairs of chromosomes, or do you have just one of the pair of chromosomes? All right, and this might make more sense after I draw this out, but for now, we're just going to talk about diploid and haploid. Where a diploid, you're going to have pairs. And then haploid, you do not have a pair. All right, again, this might make more sense after I draw this all out. Okay, so now I'm going to draw out the chromosomes of the human genome. Again, this is specific to humans. So we have 23 pairs. So I'm just going to draw pairs of chromosomes. I'm not going to draw all 23 out. We only need a couple for this demonstration, but just keep in mind there are 23. And the 23rd pair we refer to as the sex chromosome because it really determines your genetic sex. So with the sex chromosome, you could have either two X chromosomes, which are larger chromosomes that would make you female, or you can have a 1X and 1Y chromosome, which will make you male. And we call the Y chromosome the lazy chromosome because it's really just like a shriveled up chromosome. It's smaller than the X's. All right, and I'm drawing these chromosomes this way because this is kind of what they look like right before mitosis or meiosis occurs. So your DNA is going to condense into these chromosomes before that happens. And so this is not just one big, thick strand of DNA. This is a very long strand of DNA that is tightly bound to make this chromosome. All right, and they wrap around proteins called histones. And so now they're nice and tight and packed into this shape. So just imagine this as one very long strand of DNA. Okay, so now we can talk a little bit more about ploidy and what we mean by a diploid and haploid and how we have pairs of chromosomes. So let's start with pairs. Why do we consider this a pair of chromosomes instead of just individual chromosomes? And it's because they code for the same genes on each one of these. And you get these from your parents. So one of these came from your mom and one of them came from your dad. Now, it doesn't matter which one's which. We just draw them in a specific order just for demonstration, but in your actual nucleus, there is no order to which one comes first, whether it's the maternal or whether it's the paternal chromosome. All right, but again, you have two of them, one for mom, one for dad, and they code for the same genes. All right, and on each chromosome, you could find hundreds to thousands of different genes. And at a specific location on the chromosome, you're going to find a specific gene. And so we have a term for this location. We call it a locus. By locus, I mean location. So we call this location a locus, also known as loci for plural. All right, so at this locus, you will find a specific gene. Now, since you have the same genes on this chromosome as you do on this chromosome, means that you're going to find the same gene here as you are going to find here. All right, so for this locus on this pair of chromosomes, you're going to find the same gene in each one of them, right? So you have a gene here and a gene here, and in our example, we're going to say it's hair color. So that is the gene that we're talking about. Now, it doesn't have to be the same sequence here and here, though. You could have different sequences for the gene of hair color. All right, on this chromosome, you might have a sequence that codes for brunette hair. While over here, we have a sequence that codes for blonde hair. So we have a term for this. We call them alleles. So an allele is just variations or variants of a gene. So different sequences of the same gene. All right, so a gene is the trait that you're coding for, while alleles are different variations of that gene. All right, and for this, when we have two alleles, so for any gene, the most you can have is two different alleles for that gene, since we only come in pairs. So you might have the same sequence here as here. We have a term for that, and I'll talk about that here in a second. Or you might have different sequences, but at most you can only have 2 different ones for any particular gene. All right, next we're going to talk about allelic interaction. I'm not going to go too deep into this. I'm just going to use dominance as an example for everything we're talking about, because this really is just to prep you for evolution. But with dominance, we're talking about, or with allelic interaction, we're talking about how these alleles interact with each other to cause the trait that you have. And specifically, we're going to talk about dominance, which means you have one dominant allele and one recessive allele. Okay, so I just drew these out bigger to help explain this point. So again, we're gonna come here to this locus right here that is coding for hair color. That was our example. And we are going to just draw different alleles here. So I'm gonna just make this red to represent the hair color. So red is representing hair color. And let's say at this gene, you have one dominant allele and one recessive allele. So to write that, we usually use the nomenclature of a capital letter for the dominant and a lowercase for the recessive. All right, it doesn't matter what letter you choose. You could choose any symbol, honestly, but you need to be able to identify a dominant from the recessive. And this is typically how we do it. We do a capital for dominant, lowercase for recessive. And let's say the dominant here is brown hair, and the recessive trait is blonde hair. When we're talking about dominance, this means that the dominant allele is going to override or mask the recessive allele every single time. All right, so in dominance, if you have a dominant allele, this is what's going to be expressed, and the recessive will not be. All right, so in this scenario, what color hair is this person going to have? All right, so since they have a dominant allele and a recessive allele, the dominant is always going to be expressed. So what color is their hair? It's going to be brown, right? So it's going to be dominant. Dominant is going to show. Recessive is not going to show. All right, so let's look at other possibilities, though. All right, let's say this is the genes, or the alleles that they have for this gene. What is their hair color going to be? All right, so it should be brown. All right, so dominance always expressed. You have 2 dominants, you're going to have brown hair. All right, so when do we have blonde hair then? Well, there's only one case where you're going to have blonde hair. All right, the only time that you're going to have blonde hair is if you have 2 recessive alleles. There is no dominant to mask the recessive here, so that's the only time you're going to see the recessive trait. Okay, so here we have the gene hair color, right? Well, we might have other genes at other locations in your genome. So let's say way over here, I'm going to change the color up. Let's say over here at this chromosome, at this locus of the chromosome that I'm going to draw in blue, let's say this is the gene for height. All right, and let's say this person has one dominant allele for height and one recessive. Okay, so let's say the dominant trait is a tall phenotype or a tall trait, while the recessive is going to be short. All right, and since we're talking about dominance here, and this person has one dominant allele and one recessive allele, what are they going to look like? Well, they're going to be tall, right? Because this is going to be masked by this. The only time they'll be short is if you have 2 lowercase b's here, 2 recessive sequences at this gene of the chromosome. All right, so let's do one more example. Let's say we have another gene over here at this loci of this chromosome. And let's say this gene codes for eye color. this D, D. Let's say the dominant of trait is brown eyes. And the recessive trait is blue eyes. All right, so what is the phenotype? What is the trait of this person? Well, it's going to be brown eyes total because they have two dominant alleles, right? So they're going to show brown eyes. And the only time that they would show blue eyes is if they had two lowercase d's or two recessive alleles at this gene. All right, so this is going to bring us to genotypes now. So I have redrawn this genome up here just a little bigger to help explain some of these points. With genotype, let me write that real quick. When we're talking about genotype, we're talking about what alleles you have for any specific gene. All right, so we're talking about one gene at a time with genotypes. So whatever your genotype is for, let's say, this gene, you might have a different genotype over here at this gene. All right, so if we say somebody's a specific genotype, we're not going to talk about the whole genome, just one gene. Now, there's three different terms for genotypes that you will need to know when we're talking about dominant allelic interaction. And the first one is homozygous dominant. The next one is called heterozygous. And the last one is homozygous recessive. Okay, so these are going to refer to the possible combinations of alleles that you have at any one given gene. And if you know the Latin to this, this is really easy. I mean, homo means same, hetero means different. Zygous means a yoke or a bar or a joint or joining. So we're talking about what two pairs are coming together. So if you have the same pair, we're talking about the same two alleles on both the pairs. That's exactly what this is translating to. While heterozygous means different pairs, meaning the pairs that are coming together have different alleles on them. So homozygous dominant, meaning same allele of the dominant allele. So here in this case, we would have this gene be homozygous dominant, and we would write it like this. Big D, big D. Two dominant alleles for this gene. All right, heterozygous, meaning different alleles of the two pairs coming together. So out of these three examples up here, the gene of B is going to be heterozygous, meaning one dominant, one recessive. And then homozygous recessive is same alleles of the pair, but this time the allele is going to be recessive. So you see here, the example would be the A gene. So we write that little a, little a. All right, so for gene D, this individual is homozygous dominant. For gene B, this individual is heterozygous. And for gene A, this person is homozygous recessive. Okay, so let's talk about how this relates to phenotype, or what trait you're going to express. All right, so when we're talking about genotype, we're talking about what two alleles you have for any specific gene. When we talk about phenotype, is what trait is being expressed because of those alleles. All right, so just to simplify this, I'm going to make all of these A. All right, so we're just going to focus on one gene here. All right, so with an A, if it's homozygous dominant, how are we going to write that? It's going to be big A, right? And if it's heterozygous, it's going to be big A, little a. And let's say that these alleles, the dominant is going to represent, let's go back to hair color. So we're talking about the gene hair color. So dominant will be brunette or brown hair. Well, the recessive is going to be blonde. All right, so when we're talking about phenotype with the specific gene, we really only have two phenotypes. You're either going to have brown hair or you're going to have blonde hair. Those are the only two choices, right? So with phenotypes, we could use the genotypes to infer what the phenotypes are. So having brown hair. What two alleles do you need to have brown hair? Well, you need at least one dominant, right? Because that's going to display brown. And what about the second one? Does it matter? No, it doesn't matter. Because whether you have a big A or whether you have a little a, if you have at least one big A, you're going to have brown hair. So this individual will show brown hair, this individual will show brown hair. So I'm just going to leave that blank because it doesn't matter. And what about the phenotype for blonde hair? When does that happen? Well, there's only one way to express blonde hair, and that's if you have two recessive alleles. So here, this is the only individual that would have blonde hair. Okay, so hopefully you can see the difference between genotype and phenotype. With genotype, we're talking about what two alleles that do you have at a specific gene, while phenotype is what is being expressed by those alleles. All right, so in a dominance allelic interaction, you're only going to have two phenotypes. You're going to have the dominant trait expressed, where you have at least one dominant allele and the other one doesn't matter. Or you're going to have a recessive trait expressed only when the two alleles are recessive. That's the only time you're going to see blonde hair. So the last thing we're going to talk about is inheritance, because that's really what genetics is. It's the study of inheritance. And since this individual, each pair of these chromosomes came from mom or dad, so one came from mom, one came from dad. It doesn't necessarily have to be in that order. And each one of these, one came from mom, one came from dad, one came from mom, one came from dad. But they could be all mixed up. So the first one might be mom's chromosome, while the first one on this pair might be dad's chromosome. All right. The order doesn't matter. All right. Those are independent of each other. So these could be reversed or these could be reversed. All right. You might have a little B over here and a big B over here. One came from dad, one came from mom. It doesn't matter. All right, but inheritance means you have to pass on these genes to the next generation, and to do that, you're going to use gametes. All right, so I'm just going to draw. So gametes are made through meiosis, and meiosis is splitting up these pairs of chromosomes. All right, so since here in this example, I drew a female because the sex chromosome's two X's here. So one big one, so that's a female. So we're going to draw an egg. And then when this female made an egg, the egg got one of the pair of chromosomes for each of the pairs. So it got one of chromosome 1, one of chromosome 2, all right, one of chromosome 3, and so on. Okay, so maybe this one is the one that made it to this egg. Well, maybe this one is the one that made it to this egg for chromosome 2, or maybe this one made it to chromosome 3, and so forth, all the way down. All right, since we only have one chromosome or one copy of a gene, remember each one of these have the same genes on it, but now we only have one, so we only have one copy of each gene of the genome. We call this haploid. So your gametes that you make are haploid. So females are going to have haploid eggs, males are going to have haploid sperm. And then let's say some other individual male made a sperm and did the same thing through meiosis. They're going to have one, they're going to be haploid, so one chromosome for each of the pairs. When fertilization happens, all right, so the sperm and egg are going to join in fertilization. Each one of these pairs is going to match the corresponding pair in the cell that's created. So after fertilization, we create a zygote. Remember, zygote means to join. Zygote actually means yoke, like an ox yoke. I don't know if you all know what that is, but an ox yoke is a bar that holds two ox together or joins two ox together when you're pulling a cart or something. And so that's where the term came from, is to join things together. So this is now a zygote. This first cell that is made is called a zygote. All right, and this zygote is going to have one of, so let's say chromosome 1. Half the pair is going to come from the mom's egg, while half the pair is going to come from the dad's sperm. All right, same thing for 2. One from mom, one from dad, Same thing for 3, 1 from egg, 1 from sperm, all the way up to the 23rd chromosome. All right, and now this zygote, since it has pairs of chromosomes now, what is it? Ploidy. All right, now this is back to diploid. You have pairs now. Okay, so hopefully this gave you a pretty good introduction to genetics on the individual level, because now we're going to talk about evolution, which is really applying this to a population level. And in a population, you could have thousands of alleles in the population, right? But every individual in that population can only get 2 of them. All right, so remember, every individual only has at most 2 alleles for any gene. But in a population of people, you could have thousands of alleles available. All right, so some people might have a certain 2, while another person might have a different 2, okay? And in evolution, what we really mean is the changes in the frequency of the alleles in the population. All right, so before we get into the next video, just keep in mind that when we're talking about the alleles in the population, how they're found is 2 to every individual. Okay, so hopefully this helps, and just to give you a preamble into the evolution video that's going to come up next.